Hello everyone.
My name is Fabiola and my colleague
Hydeia Tyson, and we will present to you our class project, which is
Evaluation of Nutrients Availability, Mycorrhizal Colonization and Physiological Characteristics of Beans in Different Acidic Soil at Earth University in Costa Rica.
In Costa Rica, there is a constraint in beans production where the main soil limitations are low phosphorus and low pH. Every plant has its own optimum pH for growth. Changing the pH of the soil impacts on the presence of minerals which are needed dependent on the necessity of the plant. To improve the current soil ecosystem and fertility, it is imperative to supply soil beneficial microorganisms such as mycorrhizae. Mycorrhizal fungi allow plants to draw more nutrients and water from the soil. By colonizing the plant's roots as well, they also enhance plants' ability to grow and withstand acidic soil-induced stresses.
Now let's talk about the methodologies. The experimental design was a completely randomized design with the 4 repetitions per treatment for each of the 3 evaluations performed. The experimental unit consisted of 12 pots, where each repetition had the soils of different acidic pH. For all 3 treatments, the soils were inoculated with mycorrhiza and bean seeds. Soil samples were taken from 3 different areas using a auger of a depth of 20 centimeters. This was done firstly to obtain the initial soil pH and the nutrients availability in each soil. The analysis was done at the Water and Soils Laboratory at Earth University. The soils were then placed in each pot from 3 different areas. A total of 12 pots were evaluated, where we had the 3 treatments with the 4 repetitions each. The first pot was labeled Soil Casa Bodega, the second one Soil Casa Parcera, and the third one Suero Juca. After soils were placed in each pot, the beans were planted, where 8 beans were placed in each pot to consider germination variability. And then a mixture of water and 12 grams of mycorrhiza were applied to the pots with the soil and beans before and after germination. Every day the plants were watered and evaluated and analyzed for different physiological characteristics. The length and width of the bigger leaf was measured as well as the height of the stem to determine growth rate. After that, the leaf analysis was then carried out in the laboratory. The roots of each evaluations were washed and measured using a measuring tape. Now, on the observation of mycorrhiza, the roots were cut in size of 0.5 to 1.5 centimeters, then placed in test tubes covered with 10% of potassium hydroxide, remaining in that solution at 90 degrees Celsius in water bath for 10 minutes. At the end of this time, the content of the test tube were decanted and washed with hydrochloric acid and left at room temperature for 10 minutes until they became whitish. The acid was then decanted and after washed with running water. They were then immersed in trypan blue or 0.05% in lactic acid and remained for 10 minutes in this solution at 90 degrees Celsius in water bath. Then on mounting of dyed roots, after 10 minutes the trypan blue was decanted. The roots were distributed randomly in a petri dish. Slides were then prepared, placing 2 to 3 drops of glycerin that was scattered with a clamp. Segments of 10 roots per slide were mounted in a parallel, and a cover slide was placed to see the assembly and observed under microscope with magnification of 10x on eyepiece by 10x on objective. The number of mycorrhizal fields was valued as x, was then counted, that is with the presence of fungal structures such as the external mycelium, internal mycelium, spores, vesicles, and or abscus, and were divided over the total fields observed on the slide, which was valued as y. Then the percentage of mycorrhization was expressed as Z value.
Results and discussion. Based on soil analysis and the optimum levels of nutrients using the MELIC 3 for the extraction, the interpretation reflects that all the soils have low pH content. Also, all soils were presented with nutrient deficiency, especially levels of phosphorus, apart from manganese and iron that were presented in excess. Leaf, root, and stem evaluation. Though there were no statistical differences, it was observed that soils with lower phosphorus and pH content had less growth in the leaf, in the stem, and roots. This is because low pH levels affects many of the processes necessary for life, such as nutrient uptake and symbiotic relationships, where the phosphorus involved in energy transfer, photosynthesis, transformation of sugar, and nutrient movement within the plant. Though all soils have low pH and shows no significant differences between their acidity, the maximum growth yield is represented by cassava soil with a pH of 4.66, which was recorded as having the highest pH value and phosphorus level. Leaf analysis. The common beans have optimum levels for each nutrient when it is healthy. In relation to these levels, the leaf analysis from the laboratory revealed that the phosphorus uptake in the plants were low, where the optimum levels of phosphorus in beans should be 0.35 to 0.75%. There was more uptake by plants that were in soils in the plot, which is the soil parcella. The soil of the cassava, which contained more manganese of 238 parts more meson, had a much more lower phosphorus uptake of 0.19 in the bean plant. This represents the antagonistic relationship between manganese and phosphorus, where the phosphorus content decreased with increase in manganese concentration in the soil. Now we'll talk about the evaluation of mycorrhizal colonization. It was observed that all plants had content of mycorrhiza. However, the soil that was less acidic and had more percentage of mycorrhizal colonization. In comparison to an experiment related to the effects of pH on mycorrhizal colonization and nutrient uptake, it was concluded that the growth of the plant increased by increased pH and by inoculated with mycorrhizal fungi. Increased uptake was also related to the colonized root lengths, where the length of the soil in cassava was seen to be much more longer and having higher percentage of mycorrhizal colonization inside of the root. Now we will have the conclusion. The results clearly illustrated the effects low pH has on the optimum growth of the plant. In all soil analysis, there were no statistical differences between the acidity of the soil. The low soil pH results in excess manganese, which affects the uptake of phosphorus and other macronutrients. When phosphorus is limited, the most striking effects are a reduction in leaf expansion and leaf surface area, as well as the number of leaves and roots. For that, mycorrhiza are important to help increase nodulation and plant growth. Mycorrhiza help to increase plant uptake of nutrients and phosphorus, as well alleviate phosphorus stress in phosphorus deficiency soil, leading to increase in nitrogen fixation and phosphorus concentration.
After the experiment that we did, according to what we concluded, we have some few recommendations for you. It is always imperative to carry out a soil analysis test first before planting to see what nutrients are deficient and excessive in the soil. As well, perform leaf analysis tests to evaluate the nutrients uptake to see what nutrients need to be added to the soil. It is also advisable to carry out a soil and crop analysis at the end of the harvest to analyze what changed in the soil and in the crops. It is also recommended to lime these types of soil in order to raise the pH level because they are too acidic. To use fertilizers containing phosphorus in order to increase its level in the soil is a good idea, as well as applying the mycorrhiza in order to allow the plants to draw more nutrients and water from the soil, as well as increasing the plant tolerance to different environmental stresses. Thank you very much for watching.